Hello, everyone. In this unit, we will take a look at the organization of the cell to better understand the parts of the cell and what their functions are. So, a brief introduction to the cell. We've talked a great deal about the cell as the smallest and most basic unit of life. The cell is the smallest unit that can carry out all activities that are associated with life. And we know that different components or compartments within the cell work together to create a complete biological system. So just to recap, there are two major cell types, prokaryotic cells and eukaryotic cells, and they are distinguished mostly by the presence or absence of the nucleus, right? So prokaryotic cells are what we refer to as a simple cell type. There's no nucleus present, which means there aren't going to be any of the other complex membrane-bound organelles such as the mitochondria, the endoplasmic reticulum, and other structures that you'll find in the eukaryotic cell. So the eukaryotic cell by nature is a more complex cell that contains a nucleus and all of these other membrane bound organelles, right? If you were to see images of these two types of cells, it should be very easy for you to readily identify a prokaryotic cell from a eukaryotic cell. The cell theory is a unifying concept in biology that tells us that the cells are the basic unit of life, that all cells arise from pre-existing cells, and that all living things are made up of cells, right? These are three important postulates that we hold to be true, and we refer to them collectively as the cell theory. You should be familiar with the cell theory. This is a theme or concept that you will continue to hear throughout your matriculation in this biology curriculum. We know that the microscope was an important tool that basically sort of revolutionized science in such a way that it allowed us to delve into a whole nother realm of understanding life, right? There are different types of microscopes that have been developed in order to visualize different cell types. All right, the light microscope is the most common microscope that you may hear about or encounter throughout your program. The compound light microscope is typically what you would find in a basic biology lab or microbiology lab, or even like a cellular biology lab. You're typically going to come in contact with the compound light microscope. There are other more specialized types of scopes that are available for studying more fine details of cells and structures, such as the electron microscope. All right. And so there are features of the microscope that will determine how clearly we can see very small objects, okay? And so there are two features here that we're concerned with when we're using a microscope, which is magnification and resolution, all right? Magnification is the ratio of the size of the image with the actual size of the object, okay? So the magnification refers to how larger this image is becoming in comparison to the actual size of the object or specimen. Okay, the resolution then, though, refers to the ability to distinguish very fine details in an image, all right? So the resolving power of a microscope is one that we would take into account depending on what types of structures or details we're trying to look at or visualize. So things that you're able to visualize with the compound microscope are going to be basic structures on a plant or animal cell, something from human egg cells, mitochondria, so forth. Smaller objects such as viruses and so forth are going to require electron microscope in order to see these smaller molecules and so forth. Okay, so let's talk about the structure and makeup of the cell a little bit. The most important structure that we'll look at here on the cell is going to be that outer barrier, right? That protects the cell. That's going to be the plasma membrane, also referred to as the cell membrane. Okay, and so we've talked about this concept that cells must maintain an appropriate or constant internal environment. Okay, one of the ways that the cell is able to achieve that goal is by being separated from its external environment. And so the plasma membrane is the structure on the cell that's responsible for separating the external environment from the internal environment. Okay. And so this plasma membrane is essentially a surface membrane that surrounds all cells. Make a note of that, right? So we're going to, throughout this lecture here, we're going to distinguish some things that are exclusive to certain cell types and those that are common to all cell types. The plasma membrane is a surface, okay, a barrier that is going to be found on all cell types, okay? And this is an important point to note, that as we're talking about this concept of maintaining homeostasis, we need to understand that the chemical composition inside of the cell is going to differ from that outside of the cell, all right? And so this plasma membrane serves as a first sort of barrier of protection or separation between the inside of the cell and its external environment. Okay, and so we say that the plasma membrane serves as a selective barrier. Write that down. The plasma membrane serves as a selective barrier that allows the cell to exchange materials within the environment. So what types of things are we exchanging? Why is there a need to exchange, right? So we're trying to maintain and regulate this internal environment. So there's going to be a need for certain substances to come inside of the cell, right? And then there will be a need for other substances to leave the cell, right? And so that plasma membrane serves as a barrier to regulate, okay, which substances are going to be able to pass through into and out of the cell. And so then that's the plasma membrane. And so once we're inside of the cell, there are internal structures that we call organelles that are present to carry out specialized activities of the cell. Okay, so write down this term organelle. This term literally translates to little organ, right? So if you think about your body and how you're made up, you have a number of different organs in your body that all have specialized functions, right? Well, the cell has these same specialized compartments that we'll call organelles, and they carry out various activities. Okay, so let's look at this biological membrane just for a moment. All right, biological membranes are very complex structures that are made mostly of lipids and proteins. Okay, the major components found in the plasma membrane are going to be lipids and proteins. All right, these biological membranes have very vital functions for the cell. As I mentioned before, one of those jobs is to regulate the passage of materials into and out of the cell. Okay, so we're thinking about, you know, gases, water, nutrients, salt, things that need to come inside of the cell, but also waste products and so forth that need to leave the cell. Okay, that's going to happen by way of that cell membrane. Another important job of the biological membrane is to serve as a surface for chemical reactions, right? So we know that there are a number of reactions going on within living organisms constantly, and the cell surface of that membrane, the membrane can serve as a surface for these chemical reactions to take place. We know that cells interact and adhere to other cells in order to form tissues and more specialized structures. So again, this plasma membrane is going to serve as a means for cells to adhere with other cells. Okay, so we've got cell-cell contact taking place at this membrane level. And then lastly, there's communication, right, between the inside of the cell and its external environment, right? So these biological membranes serve as a means to transmit signals, right, between the environment and the inside of the cell. Okay, so signal transduction is another important characteristic of the membrane and its important role in sustaining living things. And so the structure of the plasma membrane is referred to as a phospholipid bilayer. Okay, phospholipid bilayer. This means this structure refers to the fact that there are two layers of lipids. Okay, if you look at this picture that I have here, this is a picture of a phospholipid bilayer. Two layers of lipids. We have a layer of phospholipids here, and then we have a layer of phospholipids here. All right. Now, this phospholipid is a very special type of lipid. Write down this term phospholipid. Okay, has some very important features, one being that it's an amphipathic molecule. All right, an amphipathic molecule is going to be one that contains both a hydrophobic and a hydrophilic component. All right, now this is a very important feature of the cell when we think about the job of the cell. Right, this phospholipid bilayer is going to have these hydrophilic heads out here exposed to the extracellular environment, typically an aqueous environment. And then we have these hydrophobic tails that are going to be buried inside away from the aqueous environment. So these tails are going to they're hydrophobic tails. All right. And so when you think about the membrane of the cell, this is a phospholipid bilayer. And if you think about it in sort of a 2D or 3D structure, this phospholipid bilayer will continue in this sort of circular pattern, which we can see here, where all of our hydrophilic heads are going to be facing outward and interacting with the external environment, which is water, an aqueous environment. And then the inside of the cell also, which is an aqueous environment, we're going to have these hydrophilic heads, right? And then we have our hydrophobic tails buried on the inside, okay? This again is just another illustration of the phospholipid bilayer, right? We've got a layer of lipids here, right? Hydrophilic heads, hydrophobic tails, and then we've got a layer of phospholipids down here on the bottom, okay? So phospholipid bilayer refers to these two layers of lipids interacting together to form this membrane barrier. So I mentioned before that the major components in the cell membrane are the lipids and proteins, right? We know that the lipid component here is mostly the phospholipid structure that we just got done talking about, that amphipathic molecule. Well, there are also proteins in the plasma membrane, right? The proteins are helping to facilitate some of the jobs that we talked about earlier in terms of regulating the passage of substances into and out of the cell, signal transduction, you know, we're functioning as receptors for various ligands and other stimuli in the environment. So in terms of membrane proteins here, there are two major classes of membrane proteins, which are defined based on how tightly they are associated with that lipid bilayer, right? So we have two major classes of proteins. We have integral proteins, and then we have peripheral proteins, okay? Write those terms down, integral proteins and peripheral proteins. Integral proteins, as the name suggests, are going to be those proteins that are firmly bound to the membrane, okay? Some of them can extend into and out of the cell, right? By definition, an integral protein, which is firmly bound to the membrane, would actually require that you disrupt, or disrupt the integrity of that lipid bilayer in order to remove that protein, right? So if we look at our illustration here, we have a phospholipid bilayer, right? We've got our hydrophilic heads, hydrophobic tails buried on the inside. This is our external environment. This is our internal environment. Here we have an integral protein, right? This protein, as you can see, is firmly embedded in this membrane. It extends both into and outside of the cell. And it's there in such a way that if you try to remove this integral protein, you would basically disrupt the integrity of that plasma membrane. All right. And so one example of integral proteins are those that are transmembrane proteins that are going to be able to extend all the way throughout the plasma membrane. All right, so those are integral proteins. They're firmly bound. They're in there in such a way that if you try to remove them, you're basically going to damage the cell, right? Then we have peripheral proteins, okay? Peripheral membrane proteins are not embedded into the lipid bilayer. They're typically located on the inner or outer surface of the membrane. So if we take a look here, this structure right here refers to a peripheral membrane protein, all right? This protein is embedded sort of on this inner membrane surface on the inside of in the intracellular environment. All right, so it's not firmly embedded to the entire bilayer structure. Okay, so removing this peripheral protein does not run the risk of ruining the integrity of the cell structure. And so, you know, when we're talking about the job of the cell to maintain this nice balanced equilibrium, we have to take into account cell membrane permeability. Okay. Permeability refers to um, the ability to sort of permeate or move through, right? So the cell membrane is referred to as a uh, selectively permeable barrier, right? It's selectively permeable in that not all things will be able to readily pass through the membrane, but certain substances will pass through easily and others will require assistance. So in a most general sense, the membrane is readily permeable to small hydrophobic molecules, okay, such as gases, um, oxygen, carbon dioxide. They can cross the membrane pretty easily, right? Now, water molecules are the exception here because they're, we know that these are polar substances, but they too are small enough to pass through that membrane slowly without any assistance, right? So the membrane, right, is going to be most permeable to small hydrophobic nonpolar molecules, right? The membrane though, in contrast, is going to be relatively impermeable, meaning it's not going to allow for these substances to pass through too easily. It's going to be relatively impermeable to larger charged polar molecules, all right, such as, you know, sugars, glucose, amino acids. These are larger, more complex molecules that won't be able to readily permeate this membrane without further assistance. Now, water is an exception again, because this is a polar molecule that will be able to sort of move through easily. All right. So in terms of cell membrane permeability, we're understanding that this is a selectively permeable, or some of your text may read semi-permeable. It's a semi or selectively permeable barrier that is going to dictate which substances can move in and out without too much problems. Okay. Now, those larger, more complex molecules, such as glucose and amino acids and other substances that need to cross that membrane barrier, are able to get into the cell by way of transport proteins. Okay. Transport proteins are going to be found present on all biological membranes, and they're going to facilitate the movement of needed molecules through that membrane. All right, so those larger ions, amino acids, sugars, and so forth are going to be transported into the cell, all right, by way of transport proteins. There are two main types of transport proteins, right? Carrier proteins and channel proteins. All right. These carrier proteins, as the name suggests, are going to function as transporters, right? And so what they do is they basically bind to a molecule that's trying to cross the membrane. You're going to bind to it, attach to it, and transport it across that membrane. Okay. Channel proteins, on the other hand, as the name suggests, are going to function as a channel or an opening, right? So channel proteins are going to be in the plasma membrane, and they're going to form tunnels, right, in order to facilitate the passage of molecules into and out of the cell. All right, so these channel proteins are going to create these tunnels that we'll call pores, which are basically openings, kind of functions like a gate to regulate the passage of substances into and out of the cell. Okay. The two major cell types, prokaryotic cells and eukaryotic cells, can be distinguished physically, as the prokaryotic cells are structurally more simple, right? We talked about the fact that these prokaryotic cells don't contain a nucleus. They don't contain all of the other complex structures such as membrane-bound organelles, right? So when we're comparing and contrasting these two cell types, in comparison, all cells are going to have the plasma membrane, right? All cells are going to have DNA, it's genetic material. All cells are going to have ribosomes present, right, which is the site of protein synthesis. And all cells are going to have the cytoplasm, right? This is the internal environment of the cell, right? So when we're talking about prokaryotic cells, we're mostly talking about bacteria and archaea, right? So all of the other life forms that are not bacteria, that are not archaea, are going to be considered eukaryotic cells, right? Eukaryotic cells are distinguished by having a membrane enclosed nucleus, right? Eukaryotic cells have a nucleus, and then they also have these membrane enclosed organelles that perform other specific functions, such as that Golgi apparatus or the mitochondria and so forth. Those are all membrane enclosed organelles, okay? And so again, just from a point of comparison, prokaryotic cells are going to be smaller and more simpler in structure. Okay, this is just an illustration side by side of how these two cells differ structurally. Okay, you should be able to look at pictures or illustrations or diagrams very easily and distinguish between the two. All right, as our prokaryotic cells are more simple, they don't have the membrane bound organelles, and those are usually the ones we're usually referring to bacteria. Okay, on the other hand, eukaryotic cells are going to consist of the nucleus and other membrane-bound organelles, and they are going to include everything outside of the bacteria and archaea and so forth. Okay. And so again, the prokaryotic cell is a simple cell type. There's an additional structure that's found on the prokaryotic cell, which is the cell wall, right? This is another external or extracellular surface structure that encloses the entire cell. Okay, so it's on the outside of the plasma membrane. All right. Now this cell wall it's not exclusive to bacteria, but it's exclusive to bacteria in terms of the makeup. This cell wall is mostly made up of a peptidoglycan, it's polysaccharide. It's another external structure that's going to enclose the plasma membrane. So you will find on the bacteria cell here, all cells we said are going to have that plasma membrane, which is that first barrier that separates the cell from its environment. And then we have here in the blue, which is a cell wall, which again is not exclusive to only prokaryotes, because some eukaryotes have a cell wall, right? Such as plants and fungi. Structures that are present on the bacteria cell are going to be, again, that plasma membrane, cell wall, and then some bacteria are going to have this capsule, which is just another barrier or layer of protection for the bacteria cell. Now, the eukaryotic cell is a more complex cell type. We're clear that it contains a nucleus and all of these other membrane-bound organelles with specialized functions, okay? Eukaryotic cells are partitioned into those functional compartments. Okay, the nucleus and ribosomes are going to be responsible for carrying out genetic control of the cell. That's where we're going to get our the DNA is housed in the nucleus of the cell, and then transcription and translation. Transcription occurs in the nucleus, translation occurs out in the cytoplasm on the ribosomes in order for protein production to take place. And then the other organelles are involved with sort of manufacturing, distributing molecules that are required for the cell to exist, right? And that's going to include organelles such as the endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, peroxisomes. These are all going to function in manufacturing, distribution, and breakdown of molecules inside of the cell. Okay. The mitochondria is in all of the eukaryotic cells, and the chloroplasts in plants function in energy processing, right? And then there are other structures present for structural support, movement, communication between cells. So you got the cytoplasm, or the cytoskeleton, plasma membrane, plant cell wall. These are all functioning in structural support, movement, or communication between cells. And so when we're thinking about the eukaryotic cell now, there's a point of comparison between plant and animal cells, okay? You should be readily able to distinguish structurally between the plant and animal cell. These are both eukaryotic cell types, but there are structures present in that are exclusive to the plant cell that we don't find in the animal cell, okay? Particularly this large vacuole, okay? The chloroplast. Where's the chloroplast on this? Okay, these chloroplasts here, which are going to function to harness energy for photosynthetic processes to take place. That's going to happen in chloroplasts. You're not going to find that in animal cells. All right, so I do want you to take a moment to make sure that you can structurally and functionally compare the plant and animal cell. Okay, the animal cell is going to also present with the membrane bound structures such as the nucleus, mitochondria, and so forth. All right, so you should highlight those structures and be able to identify those structures that are present only in animal cells and only in plant cells. And then I would like for you to spend some time determining or understanding the remainder of the cellular organelles that are found in eukaryotic cells. Okay, so we're talking about the nucleus, endoplasmic reticulum, Golgi complex, lysosomes, vacuoles, mitochondria, ribosomes. You should be familiar with what these structures look like and what their functions are. Okay, we know that the cell nucleus is typically, if you're looking at an image, it's going to be the most prominent organelle that you see right away on pretty much any illustration or picture, and it's responsible for housing the DNA. Okay. The mitochondria is a important structure in this eukaryotic cell where energy conversions take place. All right, so chemical energy stored in the form of ATP is going to be generated in this mitochondria. Okay, energy is produced through aerobic respirations in animal, and we'll talk about that in another chapter here. Chloroplasts are photosynthesizing organelles of plants and algae. Okay, photosynthesis, we know, is the conversion of light energy from the sun to chemical energy, to the chemical energy of sugar molecules. So glucose is a product of photosynthesis. The endoplasmic reticulum, there are two types, okay, smooth ER and rough ER. You should be able to readily identify what these structures look like and what their functions are. Okay, the Golgi complex, same thing. Come back, take some time to understand what is the purpose of this Golgi complex, which is basically to modify, package, and transport molecules and proteins. So this is where we get our glycoprotein manufacturing going on. Lysosomes, these are important small sacs of digestive enzymes, right? They're basically going to function to degrade substances and molecules that the cell no longer needs. Vacuoles are large fluid-filled sacs with various functions. You're going to see this large central vacuole in the plant cell. And peroxisomes, again, are going to be responsible for degradation. These are membrane-bound organelles with enzymes to catalyze metabolic reactions. Okay. Cytoskeleton, we're going to see these networks of fibers that's going to facilitate or sort of give the cells strength and shape and ability to move and so forth. Okay. We're going to stop here. I'm going to stop recording. I would like for you to, at the conclusion of this, to basically go back and make sure that you are familiar with the overall structure of a cell, right? Starting with the plasma membrane and working your way inside, okay? Being able to distinguish between both the prokaryotic and eukaryotic cell, all right? At the eukaryotic cell level, you should be able to distinguish between both the plant and animal cell, identifying structures that are present or absent in both. You should be able to distinguish between the roles of various organelles inside of the eukaryotic cell. Let's stop here.